Hi everyone, this is Mr. Oxler, and today I'm going to talk to you about complete dominance, codominance, and incomplete dominance. And we're really going to focus on this video just with codominance, but I'm going to review all three just so that we have a basic refresher and understanding of what they are. But the example that we're kind of going to be focusing on today is with blood types. And so we'll see how that's a pretty good example of codominance, but also complete dominance in a way. We'll see once we kind of do our little refresher. So if we look at these first two columns, where what we've talked about to start off our genetics unit, we talked about the genotype when we have a genotype where when we have a homozygous genotype where both alleles are the same, or they have the same capitalization or they code for the same thing. They have two identical alleles. Well, we're going to have whatever those alleles code for. So if we have two blue alleles, then in the genotype, then the phenotype is definitely going to be blue because that's the only option. We only have blue alleles. We only have the blue trait available. Similarly, if we have the yellow trait and we only have the yellow trait because we are homozygous for this yellow trait, homo meaning the same, they're both yellow, then obviously the phenotype is going to be yellow because that's the only option we have. Okay, but where it gets a little bit different is when we talk about a heterozygous genotype. So in a heterozygous genotype, we're going to have, in this example, we're going to have one of one color and one allele of a different color. And so the B represents blue, the Y represents yellow, and in complete dominance, what we did when we started this genetics unit, we said that one completely covers up the other. Complete dominance because one is completely dominant over the other. All right. So if blue is completely dominant over yellow, that means that this blue allele is going to completely cover up this yellow allele and pretend like it's not even there. So what do we get? Well, our phenotype ends up being blue because, again, that blue allele completely covered up the yellow allele. Now, where it gets a little bit different is when we get to things like codominance and incomplete dominance. So, unfortunately, for the fact of studying biology, it's not always so simple. Sometimes there's other traits where we might have something that where two different alleles are equally dominant. So codominance, you can think of like if you're in an organization where you have co-presidents, well, both presidents are equally in charge. So codominant means that both traits are equally dominant. So back to our first couple examples, if we have two blue alleles, well, that means we only have the blue trait available. So the phenotype is obviously going to be blue when we only have the blue trait in both of the alleles. Same thing with the yellow. If we only have the yellow traits, then the organism's phenotype is going to be yellow because that's the only thing we have. But if we have a heterozygous genotype where we have both blue and yellow, and both are equally dominant, that means that both are going to get expressed. And how does that happen? Well, it often happens with kind of like a polka dot mixture where we have some blue and some yellow, but they're both fully expressed because they're codominant. So both are completely dominant. Lastly, we also have incomplete dominance. And so we'll go back through our examples, but incomplete dominance is an example where we actually have neither one is completely dominant. So in codominance, we end up getting both that are fully expressed, and we get a full blue color and a full yellow color we get both of those in the phenotype whenever it's a heterozygous genotype. But in incomplete dominance, what's going to happen is, again, if we have only blue alleles in the genotype, it's going to be blue. If we have only yellow alleles in the genotype, it's going to be yellow. But when it's a heterozygous genotype, we're going to end up with both blue and yellow alleles. Well, and neither one is dominant. It's both of them are incompletely dominant, meaning neither one is dominant. We're going to end up getting where they mix together. So codominance, again, they're both fully expressed and they're just separately fully expressed. But in incomplete dominance, since neither one is completely dominant, they both kind of just mix together. So if you remember from art class, blue and yellow mixed together make green. So we get this green color whenever we have codominance. We get this green color whenever we have incomplete dominance and have a heterozygous genotype. So that's how genetics gets a little more complicated when we have certain traits where one thing is completely dominant and so is another thing. So this example, or if we have two traits that are incompletely dominant where neither one is fully expressed and they end up mixing together to get a new color. So here's some good definitions for that for you. Complete dominance, one allele is dominant and completely masks the presence of the other allele. Codominance, both alleles are dominant traits from both alleles. Both alleles are dominant. Traits from both alleles show in the phenotype. And then incomplete dominance, neither allele is dominant. A blend of the two alleles is expressed in the phenotype. So these are good definitions to write down. So when we move on to an example with blood types is a common example found in our body with codominance. So type A blood is expressed with this, and whenever we do codominance and incomplete dominance, sometimes we use a letter. In this case, we're using I or lowercase I, and then we put the allele in the exponent. So for type A blood, it could look like that. And the reason that we do that is, I don't really remember, but it's kind of has to do with just, you could think of it to separate so that we know that this is non-Mendelian genetics. And I kind of like the example actually up here where it has like C stands for color. And so color is what we're talking about. That's the gene that we're talking about. And then the allele is in the exponent. So we're saying, the gene color for the color is blue is the allele. Or CY, where C stands for color, Y is the color. Here, I stands for immuno something, and it has to do with I stands for isoagglutinogen. Not important at all, actually. But just think of it as the gene for blood type, and then the allele for blood type we're going to put in the exponent. So it could be IA, IB, or I blank, meaning O, because there's no antigen. So the thing in the exponent is the antigen or the blood type. It's the label that's on the outside of the red blood cells. So this one has a label that says, hey, this is an type A blood cell. This one has a label that says, hey, this is a type B blood cell. And this one has no label, meaning that it's type O blood. So if you missed my last video, type O blood has no label. So it has no antigen. All right, anyway, so when we're talking about blood types, here are the different possibilities for type A blood. We could have two alleles that are both type A blood, or we could have type A blood and type O blood. How does this work? Well, if we look at this little very, very important thing that I wrote here, which I would write down, we see that type A and type B blood are codominant to one another. They're both equally dominant. But type O blood, so type A and B are equally dominant. They're codominant. But type O blood is less dominant than either one of these. So type A blood is completely dominant over O. Type B blood is completely dominant over O. But type AB blood are equally dominant. So when we look at this again, we have type A and type A. Well, obviously that's going to be type A blood. But if we have type A and type O, remember, A completely covers up for O, and we end up with type A blood still. And type B blood, we have this one would obviously be type B blood. It's the only thing that both alleles are type B. That's the only thing it could be. Here, when we have heterozygous for type B and type O, again, type B completely covers up type O, and you end up with type B blood. For type AB blood, the only way we can get type AB blood is AB blood represents both alleles in the genotype. Or on a blood cell, it represents type A and B antigens on the surface of the blood that label them and say, hey, this is type AB blood. All right, so it has both of those antigens. So in the genotype, it's going to have an A allele and a type B allele. Type O blood, as I said before, type O means no antigens. All right, so when we draw our little eyes, that again kind of signal, it stands for isoagglutinogen, and it's kind of saying, hey, here is the blood type. Well, it has nothing there because there is no antigen. So the only way we get type O blood is when we have I with no exponent and I with no exponent, and there's type O blood. Otherwise, if we have an IA with an O, again, the A covers up for the O. So that would be type A blood. All right. So here's our key with kind of our different options, or I should probably circle the whole thing. And then this is a very important key here that says, again, type A and B are codominant. They're equally dominant to one another. Type O blood is going to not have any antigens, so it just doesn't have an exponent. And again, type AB blood are going to be equally dominant. Type A is dominant to O. Type B is dominant to O, but type AB are equally dominant to one another. So let's do a couple of examples of some blood type crossing using Punnett squares. So if somebody with type A blood, they fall in love, they get married, they have kids with somebody who has type B blood, and both people are homozygous for these blood types. Well, the only thing the kids could have is, if I don't even do the Punnett square, I could look at this and say, well, this person has a 100% chance of giving an A allele because that's all they have. This person has a 100% of donating that B allele to the child. So that's because that's all they have. I can plug it into the Punnett square if I want, but we're going to end up getting 100% type AB blood. All right, our next example, well, what if somebody has type A blood and they marry somebody who has type A blood also? But both of these people are heterozygous. Both of them have an A allele and an O allele. Well, what happens? Well, let's see. Let's use our Punnett square. So The first option is they could have kids that are homozygous for the A blood type. We could have kids that are heterozygous for the A blood type, just like the parents are. We could have the same thing here where the kid is heterozygous for the type A blood, or the kid could actually have type O blood. So if you watched my last video where I was talking about how blood types are an easy way to, and not necessarily a definitive way in some regards, not others, but blood type tests are kind of the original paternity test. If one parent has one blood type and another parent has a different blood type, sometimes it's impossible for that kid to have some other random blood type. And sometimes it means that one partner cheated on the other partner, and so it can lead to some fights. But here would be an example where the kid has a blood type that neither one of the parents have, but it could still be their kid, so they don't need to freak out. So again, learn genetics before you end up filing for divorce for no reason with your future partner. All right, so here, let's do a couple more examples. Well, what if we have somebody with type A blood and they're heterozygous for it? So why do I keep drawing that? So they're heterozygous for it, so they might look like that. And then somebody else that's homozygous for type O blood, or in other words, they have type O blood, and that's the only way they can have type O blood. Well, when we plug this in, we get that half of the kids are going to be type A, and the other half are going to be type O. And then here's another example here. Well, what if we have type A blood that's heterozygous for it, and the other partner is type B blood, and they are heterozygous for that? Well, what could the kid have? The kid could be AB. The kid could have type B. The kid could have type A, or the kid could have type O. All right, so we can do some more examples with that later and see what would happen in different scenarios. But again, this kind of gives you some insight on how these very basic paternity tests work, and it's kind of a cool thing to know how to do. And then it'll help you when we have a test over stuff like codominance, because it's a very real life example of codominance. Again, codominance means that we don't get any kind of mixture. If we have type AB blood, like this person or any of the kids in this example, they don't have like some sort of half A antigen, half B antigen. No, they have their red blood cells have a full type A antigen and a full type B antigen. The type A antigen is fully expressed and it's fully available on their red blood cells, and the type B antigen is fully expressed and fully on their red blood cells as antigens. So I hope this was helpful. If you have any questions, leave a comment below, and I'm happy to help you out.